The shape of your cell determines what you are capable of being. It is not destiny, but structural geography. Imagine your DNA isn't a fixed scroll, but an enormous, impossibly complex three-dimensional city constantly shifting its architecture. This physical folding dictates which parts of the code can even talk to each other, the regulators and the genes themselves. When you experience deep focus or intense stress, tiny shifts happen in that urban layout. These changes are not just chemical markers, they are reorganizations of chromatin topology itself. It's how your quietest mental states begin subtly restructuring foundational genomic pathways, changing who you might become without touching a single base pair of DNA.
It is not enough that these shifts happen. We need to see how they operate on a cellular level. Think of your nucleus as being divided into functional neighborhoods, areas called topologically associating domains, or TADs. These are stable regions where specific genes communicate with their dedicated enhancers. When you introduce patterns from deep internal rhythmicity, like consistent metabolic oscillations during intense focus, that signal biases the structural integrity of those domains. It doesn't just flip a switch, it subtly shifts entire neighborhoods within the nucleus. The cellular machinery then reorganizes itself to reinforce this new arrangement, essentially building a stable physical map for a different kind of self.
But the reorganization isn't just random, it's regulated by chemical command signals. Consider acetyltransferases and deacetylases as microscopic gatekeepers. They are enzymes that modify the histone proteins, those spools around which your DNA wraps. These modifications dictate whether a gene neighborhood is open for action or tightly silenced. What we're finding suggests that sustained internal states, like periods of profound calm coupled with deep attentional focus, don't just.
This is where the map meets reality. These microscopic physical shifts aren't just abstract changes, they directly mirror processes we know govern long-term memory and neuronal plasticity. When our chromatin structures rearrange themselves to support a new functional state, this precise structural change correlates with the way stable memories are physically encoded in neural tissue. Think of it like carving permanent grooves into concrete. The rearrangement is not temporary drift, but an architectural commitment. The physical topography becomes information storage itself, validating that profound subjective experience can leave measurable genomic residue. But here's the complication. While these studies show correlation between state and shift, a deep reading suggests some researchers argue this architecture might be entirely driven by random metabolic noise rather than directed intent.
The idea of randomness is compellingly simple, but biology resists such neat conclusions. If all structural shifts were pure noise, we would have no consistency in self-transformation, no pattern at all. We are moving past random fluctuation and into signal transduction. Think of the cell's metabolic state not as background hum, but as a sophisticated interpretive language. A specific dietary or stress condition can activate certain cellular pathways that act like finely tuned biological filters within the nuclear environment. These conditions bias how the chromatin machinery responds to subtle rhythmic shifts in our internal states we discussed earlier. The external signal gets interpreted, but only when it reaches this crucial gatekeeper pathway does the cell decide which architectural shift is stable enough to hold on to forever.
The most challenging leap is reconciling intent with biology. If our subjective experience drives a physical shift, does that mean we are merely steering an already deterministic ship? The deeper insight shows it's not just one way of moving. The system operates in cycles. Neural activity doesn't simply command chromatin architecture from above. Rather, sustained patterns of high attentional focus create specific metabolic oscillations within the cell itself. These internal ripples then feed back and amplify certain structural configurations into lasting domains. It is a self-reinforcing biological loop. Your pattern influences structure, and that new structure biases future input, creating genuine emergence. But this suggests we are not merely editing a script, we are actively writing its operating code while the cells read it in real time. Does total control exist when every shift creates a dependency?
We are not just passengers in this system. We function as dynamic input biases. The final realization is that agency does not demand absolute command over structure, but rather precision regarding internal states. When you align deep attentional patterns with specific metabolic rhythms,
whether through structured breathwork or sustained mindful focus on a singular quality of presence. You are sending the clearest possible signal to those enzymatic gatekeepers we discussed earlier. You aren't forcing change. You are optimally priming the cellular machinery to stabilize favorable architectural outcomes. This is self-design via subtle biological instruction set, rather than sheer willpower, leveraging complexity for directed emergence. The full research archive is at jmthomasofficial.com.
This is the ultimate convergence of biology and philosophy. We have seen that matter organizes information, structure creates function, and intention becomes a physical force through biochemical translation. The self is not an isolated entity governed by fixed code. It is rather a constantly negotiated state, an emergent property defined by its interactions with internal rhythms and external conditions. This realization dissolves the strict boundary between mind and material world into a single complex operational system. We are, biologically speaking, continuous pattern generators whose architecture reflects our deepest modes of being. The true revolution lies in accepting this fluidity over rigid determinism. Your state is not just observed, it structures reality at its most fundamental level.